Hello everyone. Today I'm going to discuss about the central dogma of molecular biology. Let's start with the DNA replication. DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. Each newly synthesized molecule contains one parent template strand and one new daughter strand. DNA replication is composed of three major steps. Step 1 is the initiation. Each helicase unwinds and separates the DNA helix into single-stranded DNA. As the DNA opens up, Y-shaped structures called replication forks are formed. Next is the step 2, the elongation. During the elongation, an enzyme called DNA polymerase adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strand. All newly synthesized polynucleotide strands must be initiated by a specialized RNA polymerase called primase. Next is step 3, the termination. It occurs when two replication forks meet on the same stretch of DNA, during which the following events occur. Although not necessarily in this order, the forks converge until all intervening DNA is unwound. Any remaining gaps are filled and ligated. Catenanes are removed and replication. The next is the transcription. Transcription is the process of RNA synthesis, controlled by the interaction of promoters and enhancers. Several different types of RNA are produced, including messenger RNA or the mRNA, which specifies the sequence of amino acids in the protein product, plus transfer RNA or the tRNA and ribosomal RNA, the rRNA, which play a role in the translation process. The next is about the transcription. Transcription has also has steps. First step is the initiation. The DNA molecule unwinds and separates to form a small open complex. RNA polymerase binds to the promoter of the template strand. The next step is the elongation. RNA polymerase moves along the template strand, synthesizing an mRNA molecule. In prokaryotes, RNA polymerase is a hollow enzyme consisting of a number of subunits, including a sigma factor that recognizes the promoter. In eukaryotes, there are three RNA polymerases, 1, 2, and 3. The process includes a proof-feeding mechanism. The next step is the termination. In prokaryotes, there are two ways in which transcription is terminated. In row-dependent termination, a protein factor called Rho is responsible for disrupting the complex involving the template strand, RNA polymerase, and RNA molecule. In row-independent termination, a loop forms at the end of the RNA molecule, causing it to detach itself. Termination in eukaryotes is more complicated, involving the addition of additional adenine nucleotides at the 3' of the RNA transcript, a process referred to as polyadenylation. The next step is about processing. After transcription, the RNA molecule is processed in a number of ways. introns are removed and the exons are spliced together to form a mature mRNA molecule consisting of a single protein coding sequence. RNA synthesis involves the normal base pairing rules, but the base thymine is replaced with the base uracil. Next is the translation. In translation, the mature mRNA molecule is used as a template to assemble a series of amino acids to produce a polypeptide with a specific amino acid sequence. The complex in the cytoplasm at which this occurs is called a ribosome. Ribosomes are a mixture of ribosomal proteins and ribosomal RNA and consist of a large subunit and a small subunit. The first step of translation is initiation. The small subunit of the ribosome binds at the 5' end of the mRNA molecule and moves in a 3' direction until it meets a start codon, the AUG. It then forms a complex with a large unit of the ribosome complex and an initiation tRNA molecule. The next step is the elongation. Subsequent codons on the mRNA molecule determine which tRNA molecule linked to an amino acid binds to the mRNA. An enzyme, peptidyl transfer, links to the amino acids together using peptide bonds. The process continues, producing a chain of amino acids as the ribosome moves along the mRNA molecule. The next step for translation is the termination. Translation is terminated when the ribosomal complex reached one or more stop codons. These are UAA, UAG, or and UGA. The ribosomal complex in eukaryotes is larger and more complicated than in prokaryotes. In addition, the processes of transcription and translation are divided in eukaryotes between the nucleus and the cytoplasm. The nucleus is in the transcription and the cytoplasm in translation, which provides more opportunities for the regulation of gene expression. And that is all. Thank you.